Now you can review that. I was going to do a poll everywhere. How old is Dr. McFarland? But somebody's going to get their feelings hurt one way or the other. So I'm not going to do that. But you're welcome to guess in your own mind and be wrong. Okay. So, but the other thing is, because it's my birthday, I'm actually going to take some time to talk about the stuff that I really like to talk about for a few minutes, because it was at the end of the last lecture, so I put it off to this one. For this first group of lectures, remember it's mostly review, and so there's going to be some stuff that you're like, what am I talking about? And some stuff that you're like, oh yeah, I remember that. So your job for this first test is to sort of go through that stuff. For the stuff that you're lost on, go back and listen to the recordings. That's why I'm recording it, so that you can go back and get that in more detail, do searches around it, and then if you can do the homework problems that are related to it, and if you can understand the slides, then you've got it. You know, that's what it comes down to. So we'll talk about that, but you're going to have to do some sort of like reconnecting to your past knowledge. And everyone has some gaps in their past knowledge, and this first part is about filling in those gaps in a biochemical sort of big picture type thing. So, with all that in mind, I would like to make some connections, not just to science, but to theology as well. And maybe this will give you something to say, to talk about, to ask your theology professor about. In fact, today I'm going to mention both Origen and St. Augustine by the end of it. Now, because of this, I'm already giving myself up that this lecture is going to run over today. I'll let you go at 12:20, but then I'll probably record the rest of it. So we're going to have like I promise not to do this more than 1 or 2 times. I might have to do it 3 times for this test. But I promise for the future tests, I'll try to keep it down to where we have these post-lecture add-ons. And I'll always try to make them to be less about the discussion, more about like the, oh, isn't this cool kind of additions to what you already know. With that in mind, please ask questions and discuss, because we will make time for it using the technology that we have. So one of the ways I would like to make time for it, so I'd like to talk about the theology of homeostasis, okay? If you just use that word, if you understand that word, you can actually use it in every single class on campus, I think. Maybe not math. Well, even math, okay? So talk to me about that. But the thing about, you can definitely use it in chemistry class and biology class, is the fact that you have the atoms in something changing. So at the very lowest level, we think of everything as being built off of the lowest level, right? But the lowest levels of everything is in flux. I'm not even talking about going down to quantum mechanics. I'm talking about going down to the chemistry, to the atoms that are moving in and out. Whenever you have atoms moving in and out of you, technically your matter is changing. But matter is not the only thing that matters, okay? I have to do one kind of joke like that every single time. But what matters to the fish persisting is actually moving matter in and out of the fish. So how do we describe this? This fact that the fish's atoms are dynamic, but the fish itself remains static. It remains the fish. It retains its identity. So the thing is, the chemical arrangements of the fish are retained even if the atoms are not. So realize what's important here is not the atoms, but the relationships between atoms. This is relational. Again, we're starting to get into theology now. And homeostasis shows up all over life, at all sorts of levels. Whenever you have something being maintained against the pole of equilibrium and entropy, you have homeostasis. And it's really important to point out, homeostasis is not equilibrium. In homeostasis, you have something alive that does not appear to change. In equilibrium, you have a chemical system of 2 reactions that are opposing to each other that don't appear to change. So they are both not appearing to change, right? But you have, for homeostasis, you have the arrows pointing in different directions. Equilibrium, what's equal is the rate of the forward and the rate of the reverse reaction. So you have 2 arrows diametrically opposed between 2 states. Homeostasis, you have the thing that's not changing in the middle, and it's taking in, you have the arrows like this. So this is homeostasis, this is equilibrium, okay, if my hands are arrows. So homeostasis can happen with hemoglobin, and it also does involve the rates. If you have all of the rates spelled out, and if the thing in the middle is staying the same, then you know that the rates are equal. the hemoglobin staying the same, and it's coming from amino acids that are being synthesized to hemoglobin, and then each individual molecule might last for a couple of days and then get broken down. But overall, your hemoglobin levels are staying the same. You go into the doctor, you measure your hemoglobin. Okay, it's where it was before. Good, you're healthy. What this means is not that the hemoglobin is staying there. What it means is that the hemoglobin is being made at the same rate that it's being broken down. It means that R1 equals R2. Notice these are rates, not concentrations. So what's equal here is the rates. And actually, that's the same thing for equilibrium, but the rates in this case are arranged in a line with the state that is staying in the same is in the middle. So you can also say blood glucose should be the same. You know, if you don't have a diabetic condition or something like that, I mean, it'll fluctuate a little bit. Like you'll be hangry, right? And you'll eat a Snickers or whatever that thing is. But you're trying to regulate this, and you're trying to keep it within certain bounds. And again, a healthy individual will have mostly the same glucose levels. Food has carbs that go in. The glucose is used in several different ways. It can be breathed out as broken down waste. It can be stored as fats, or it can be turned into other things. It can be turned into ribose for making DNA. So the thing is, if the glucose is staying the same, if you have all the rates, in this case, R1 equals the 3 out rates. That 1 in rate will equal the 3 out rates. This is actually quantitative, and so it's kind of cool. At this level, we can talk about homeostasis, and it shows up all the way in medicine. So future doctors, the fact you want to keep your patients in a state of homeostasis because you don't want them to die. You don't want your patients to reach a state of equilibrium. So you're fighting against equilibrium. The Doctors Against Equilibrium could be a biochemical organization I could get behind. So the thing about this is that if you have the iron kinetics, iron can be in 4 different places in the body, and you can draw an arrow between each of these, and it can go in certain directions, and you know that the arrows, the amount leaving the plasma is 35 mg per day. The total entering the plasma should be 35 mg per day, and it can be divided up in different ways. But the important thing is that these 2 rates are equal. If the rates in and the rate out is equal, then you have a case of homeostasis if it's more complex than just 2 states pointing at each other. Chemistry is this, biochemistry is this, okay? So pay attention to what's equal here. And if the equality gets out of whack, then you have an unhealthy state for the organism or patient. This holds the case for proteins in general. In general, proteins are made through the central dogma, right? They're transcripted, they're translated, and then you have the unfolded protein, and then it's supposed to fold. Well, we have a whole chapter on folding coming up, so I can tell you that it's really complex, and you have a bunch of reactions, but you can break it down to the reactions, and you can say that you can have all these different folding reactions, but when you step back, the amount of folded functional protein tends to stay the same in an organism because the organism tends to stay the same. The proteins are dynamically changing and undergoing all these different reactions. What matters is when you step back, do all the reactions result in the functional protein maintaining its concentration, the same amount of the protein that's doing its job. And you can have protein that is non-functional and protein that is even pathological. It can cause disease. We'll talk about all that, but the thing about each of these is a chemical reaction. And if you step back from this is a cycle, and if any one of these components is maintaining the same, let's see, where's the functional? Here's folded functional protein. If this is staying the same, then what you know is that the rates in and the rates out are going to be equal. But you don't know that they're going to the same place. And so we have all of these cycles within cycles. And this is just the protein cycle. There's cycles for the iron, and we've talked about cycles for an individual protein, hemoglobin. All those are in, and I like this one because it sort of looks like an infinity sign. That's just the way they arranged it. The important thing is that you have closed circles at a certain level. These are not equilibria. Like this right here could be considered an equilibrium, but this is not an equilibrium, is some kind of cycle or circle, and that's where you can have homeostasis for one part of it. But you can zoom out too. You can zoom out to the entire ecosystem. So the fish is in a state of homeostasis. The fish dies, it's no longer in a state of homeostasis. The fish has higher concentrations of salt inside it than outside it. Let's say it's a freshwater fish, right? And so you have these concentrations in the fish of the salt ions are higher than the concentrations in the lake. When it dies, it's no longer maintaining that homeostasis, it's no longer fighting against equilibrium and entropy, and the salt spreads out. It spreads out from inside the fish to become in equilibrium. Here is where the concentrations start to equalize with the environment, okay? When it reaches equilibrium, where the concentrations are equal to the environment, but more importantly, the rates of moving to the environment, moving in and out of the fish are the same, then you end up with the concentrations being the same inside and outside the fish. The fish is no longer alive because it needs those ions to live. It's already died and it's releasing those ions now. But even more than that, more than the ions being released, bacteria start to gnaw on it and they start to break it down, and eventually the fish disappears to our eye. But we know conservation of energy, conservation of matter. The atoms that were in that fish are now in little bacteria, and they've been broken down to their components, like we say bacteria will do. They get energy from spreading things out. So if you break stuff down to its monomeric subunits, the stuff that you memorize, or if you even, bacteria will break it down even more to phosphate, CO2, and NH3. These tend to be the more stable components. The smaller it is, it's a general rule of thumb, and not an absolute truth, but these tend to be the most stable molecules. And then bacteria start to use these to build themselves up again. Fish eat the bacteria, big fish eat the little fish, and then you can have a new fish, okay? That's also in a state of homeostasis. But step back from this and look at the whole ecosystem. If the ecosystem is stable, you have another case of homeostasis that applies at the system level. If you have the same number of fish when you go back, if the same number of big fish, same number of little fish, same number of bacteria, you don't have an algal bloom or things like that, then you can have an ecosystem that is also dynamically changing. The fish are eating, the fish are putting out waste, some fish are dying, some fish are living. But if you come back to that, it's like the iron pool in the body. It stays, the concentration of fish stays about the same, and you wouldn't say that the concentration of the amount of fish in the ecosystem is pretty clear. That's a dynamic system that's constantly changing, but it's stable in its dynamic nature. That is the characteristic of life. And so this follows the rules of thermodynamics as well. Just realize that the fish holding itself together has to pay a cost in entropy as it lives in spreading out the food that it eats as for more spread out waste. And it gets the energy in terms of delta G. And that's why we talk about delta G, delta H, and things like that. We're bringing those back, and we'll use those concepts throughout the course. So the smaller molecules are more stable. The fish's life reactions are no longer maintaining the biopolymers when it dies, so bacterial enzymes start to do it. And this is a chemical reaction. These are enzymes breaking bonds. And so we can characterize this chemically. So cold temperatures, like most reactions, if you make it cold, it will happen less. And that's why you put fish on ice. Because if you don't put fish on ice, what happens to the fish? What molecule do you start to detect if you just leave a dead fish out on the deck of a boat? Anybody? What's that? Yeah. So, oh yeah, I have it down here so you can read, right? But you'll start to smell, the fishy smell is ammonia. And in the lab, by the way, if you're ever working with an amine-containing compound, it smells like fish. Because fish have a lot of amino acids. The amino acids become ammonia as bacteria break it down. Another method is to make fish jerky. Okay, so you can dry the fish out, and it won't break down nearly as fast. Why does drying the fish out help keep it from breaking down? What do you think?
Is it because a lot of degradative processes or hydrolysis reactions that require water?
They require water at a certain level. They could require water at the direct level. The hydrolysis of amino acids actually requires water at the direct level. Or they could require it independently. The bacteria require water just to live. So you're kind of starving out the bacteria from the water that they need. And if you just remove water, you can preserve something. The enzymes can't move or react if you don't have water. And this is very true for the ones that use water as a reactant, but it's true for enzymes in general. Enzymes assume that they'll have a homeostatic pool of cytoplasm to work in. They wouldn't put it that way, but you can put it that way, okay? They need homeostasis. And so the thing is that you can get all sorts of crime dramas actually are related to this, because you can talk about what if it's, we talk about humans, fish are made of atoms and humans are made of atoms too. And so if you have like a crime drama, you can actually measure the divergence from homeostasis, the way in which things are seeping out like the salt seeped out from the fish, okay? One of the ways that you can do this is by looking inside the eye. The vitreous humor inside the eye, post-mortem, will start to fill with iron, magnesium, and molybdenum. These are supposed to be inside cells, but if the cells are starting to die, they're leaking out, and they leak out into, they let go of their precious iron, and it goes into a place where you can measure it. So the cells break down, the metals leak out, and so you can measure the metals with mass spec, and you can determine the time of death from that. This is so this is one way you can do it. I want to show you how you can actually do it with another bodily system. The same thing works in the bloodstream. You get iron released into the bloodstream just like you get it released into the vitreous humor. And another thing you get released into the bloodstream is urea. That's another breakdown product that should be not in the blood because it's actually a dangerous chemical. That's a chapter way down the road about the urea cycle. But the main thing is, should not be in the blood, but after something dies, it's reaching equilibrium. The concentrations outside the blood and inside the blood are beginning to equalize. So what they did is they actually looked for what kind of things end up in the blood after death that aren't there before, and they measured, over 24 hours the things, the positive slope right here, iron, urea, and certain enzymes. Much later, they'll talk about why these enzymes are there, but the most important thing, iron should be inside red blood cells. It should not be outside. After blood, it starts to follow this line, and it starts to be released into the blood. And you can actually measure the concentration of iron and the other things, and you can say, oh, the concentration of iron is 100. Therefore, this person died 250 hours ago, and we can put the time of death, and we can look at the motive, and we can look for the murderer, okay? This is actually kind of a CSI kind of thing. So this works for fish, it works for CSI shows, it works for humans, it also works for wildebeests, okay? So we're going to talk about the paper that we're going to assign, that we're going to read through. It's an ecology paper, actually. Dr. Long would be proud. And we're going to have Serengeti wildebeests. I guess this is kind of in honor of the Lion King. Maybe I should see that movie. Anyways, but they have nutrient cycle. It's circle of life. And so I totally just realized this is a Lion King paper. Maybe we can assign a movie to every paper. But the thing is, every part of the ecosystem plays a part in keeping and making it stable. And how this works is really amazing sometimes. The large herbivores, the wildebeests, actually stabilize the food web as part of their whole annual mass drownings, So this is actually bad for the wildebeest, but it's good for the ecosystem, okay? And we can talk about where it's good on an elemental level, on a chemical level. So this is chemical ecology. Again, Dr. Ferrer would be proud, okay? So this is, start looking at this paper. I put it online so that you can start reading it through. Everything for this first one, work a little bit on it every day. Get stuck, come back to it. And it's like working on a crossword puzzle for me. Like I get stuck and then I come back to it like an hour later and all of a sudden I'm like, oh, that makes sense, it comes to me sometimes, not always, yeah.
So what's the end goal with the reading? Is it like a set of
Mhm.
questions with it, or is it just for...
On the review day when we bring everything together, the end goal for the reading is for us to be able to discuss the paper. So, and then we will be tested on the paper, but we'll have a class discussion where we all bring the paper. I like to print it out. You can do whatever you want with it, but you're supposed to bring the paper in some form to the review session a week from today.
Okay. And the stuff we're reading from the paper is going to be on that version?
Yes.
Okay.
Yes. So Test 1 review day will include all the stuff for test 1, and that's when we'll talk about the paper most. Yeah.
Can we have the printout at the exam?
No. But I will give you parts of it. The reason is I will give you one part of it, but I'll take out part of it. From the part that I give you, you should be able to read, if you can read it. So I'm testing your reading comprehension. I'll give you part of the paper on the exam, and it leads to conclusion A. So you should be able to come to conclusion A if you just remember, if you can read the paper. So that's what I'm really testing. I'm testing if you can read the paper. So I don't want you like memorizing the numbers. I want you memorizing the reasoning. We'll talk about all that next Friday. And this is a good starter paper, by the way, because it does just involve these concepts in the lecture 2 that we're talking about mostly. So you can actually go ahead and start it right now. And so like do it bit by bit. Just like you have to eat with little bites, you know. You don't eat all your food in one day. You don't learn everything that you need to learn in one sitting, okay? Cramming is also kind of like eating too much food. So it's, that metaphor breaks down at some point. But what's that? Yeah, so is the food. There you go. So, but the thing about this is that don't identify yourself too much with your atoms. Don't hold on to your atoms because you're losing them all the time. You're shedding atoms in several different forms of waste. You're breathing out CO2. So every 5 days, your stomach lining turns over. Every 2 months, your liver turns over. Your skin replaces itself every 6 weeks. There's some movie where they go onto an old abandoned spaceship, and it's all dusty. And they're like, why is it so dusty? Well, it's because the human skin turns into dust. Most of what you see as dust is actually human skin. And so they had a very dusty because they had an abandoned, and they had humans that were breaking down inside the ship. No spoilers, but that's how you can get a dusty spaceship, okay? Your skin turns into dust. Your liver turns into atoms, okay? Dust to dust. 98% of the atoms of your body are replaced relative to last year. And yet here I am asking you to remember stuff that you learned last year in organic chemistry. Is that fair of me? I think it is, because you have your mind and your soul that have actually been maintained throughout that period. So it doesn't depend on the atoms. So I want you can think about this. With this insight, you can think about this, and this is not really that new of an insight. People have always known that you eat stuff, you poop stuff, you know, stuff goes in, stuff goes out, but you stay the same. You sort of age and get old much more slowly. So even the theology of the resurrection, that is a physical discussion. It talks about the body being raised. And if I talk about resurrection, you can go back not just to the Gospels, but you can actually go back to Ezekiel, and you can see Ezekiel looking at the valley full of bones that come together. This is very physical language at the very least. What is Ezekiel saying here? Well, at the very least, he's not presenting it as a clear metaphor. There's clearly something very physical going on here. A noise, a rattling sound, the bones come together, tendons and flesh and skin. This is about as precise as Hebrew can get when it's talking about anatomy. So we're talking about Ezekiel using this, and at the same time he's talking to the whole nation of Israel. How does that work? Well, go into theology class and think about how Jesus summed up the whole nation of Israel, but that's another topic for another day. But this touches on that. I just want you to see, if you're talking about life, you're talking about things in all these different classes. So if you have the Christian theology and promise of a bodily resurrection, what does that mean in terms of atoms? Will this atom right here be with me in the resurrection? Okay? Well, probably not. And does it really matter? But will atoms be with us at all? That's a more interesting question, and it's more debatable. You can talk about all this, but I wouldn't exclude the idea that atoms would be with us at all. The new heavens and the new earth could be atomic in nature. Let's look at the evidence and think about it. What is the thing that will be with us? What is the thing that, when Jesus said to the thief on the cross, "Today you will be with me in paradise"? Okay, there's something that was being persisted and that was being promised in that case. What is that? It's not the atoms, at least it's not most of the atoms. You know, so really interesting things to think about. Realize that these are things that are old questions. Science has been talking about those for a long time. But the fact that we now know more detail about the tendons and sinews does not negate what Ezekiel said about them. Okay? People knew that dead people stay dead. And so the resurrection, whatever it means, is going to mean something in that context. So it's important to realize that with science, we have found out a lot of new things. And when we gain new knowledge, it's tricky to sort of put it into to say what does this mean in terms of an old language and framework. But I don't want to just make it wave the problem away by saying that science is all there is, and therefore we have to take everything as metaphor. Very clearly in the Gospels, the resurrection is not presented as a metaphor. It's presented as a bodily act, including the ingestion of food, which is a homeostasis-like involved event. Like I said, these are old questions. There was actually an old debate between Origen, one of the church fathers, and Celsus, who is a Roman skeptic, in the 3rd century AD. And the skeptic asked him a challenging question. Okay, what if you have a missionary going to a cannibal, and the cannibal, the missionary preaches to the cannibal, but then the cannibal eats the missionary? Okay? Some of the atoms of the missionary become the atoms of the cannibal. And then, because of the example of the missionary, or maybe because of God's grace entirely, the cannibal becomes a Christian. In the resurrection, who gets whose atoms? You know, it's actually kind of an interesting question, but it really is along the line of the Pharisees challenging Jesus. I don't think it's a real serious question. But Origen said, you know, God made the atoms. God's putting them back together. God knows what's going on with that. And also he said, we're in constant flux. So I don't own my atoms. You do not own your atoms, although the body is also redeemed, you know. Spirit and soul have always been represented by life-like processes. And if you look at the terms used, they're dynamic, they're dissipative, which means they talk about things like blowing places and the something's coming out of them that is more spread out. Emergent and irreducible cycles. So like wind and fire are processes, not elements. Now we know that fire is not an element, but that actually makes it a better picture for spirit than if it was an element, you know? We can't purify out the spirit with chemical means. Processes, not elements. This means that the self is ungrounded, at least ungrounded in atoms. I believe that the self is grounded in God as a gift from God, but that's, again, theology. I've got to get back to biochemistry one of these days. But the prospects for life after death, the content of Jesus' promise to the thief, has to remain imponderable and unamenable to empirical determination. You cannot redo the reaction of dying for yourself. You know, you can die to yourself, and we can get into all that. But the thing about death is irreversible, and you can only undergo that once from your own perspective. So what happens to my perception after I die? Well, I'm not going to say that science has really much to say about that. It's not a repeatable experiment that you can do. So, with all that, that's my birthday theology lecture. Let me actually